Hi everyone, it's Mr. Sinti, and today I'm going to talk to you about the classic process of translation, which is the second half of protein synthesis. It's where messenger RNA, ribosomal RNA, and transfer RNA all come together to make a protein or a polypeptide. And it happens on a ribosome in the cytoplasm. So I want to start off with just a little drawing here of the cell, just to give you some orientation. So here's a typical eukaryotic cell membrane. Here's the nucleus. Let me create some pores here in the nuclear membrane. And as you know, DNA resides in the, as you know, DNA resides in the nucleus. And so here it is residing in the nucleus. And it's copied into a smaller molecule called RNA, and that's called transcription. Well, as it turns out, messenger RNA is going to leave the nucleus, and it's going to go out into the cytoplasm, not drawn to scale, where there's many ribosomes. And so the messenger RNA is going to go to a ribosome, and here on the ribosome, the messenger RNA is going to be translated. In other words, the nitrogen base sequence is going to be translated into an amino acid sequence like this. And so this little guy coming off the ribosome is, in fact, you guessed it, a protein. And so protein right there. And so what we're talking about is this process. How is the genetic code read or translated into a polypeptide? And that occurs on a ribosome in the cytoplasm. And so let's get right into that conversation if we can. Let's go back here, and let's go here, and then here. And so this is from a previous discussion that we had, that located on different segments in DNA are genes, which are nitrogen base sequence that code for the polypeptide, i.e. the amino acid sequence of the protein. So here's the gene, and when that's transcribed into messenger RNA, the messenger RNA is complementary. It's single-stranded. And again, from our previous discussion, we know that the genetic code is read in groups of 3 nitrogen bases. So it's 1, 2, 3 is a codon. And that corresponds to one of the amino acids, and then 1, 2, 3, another one, 1, 2, 3, another one, 1, 2, 3. And so how do you read the genetic code? You read the genetic code based on the decoder. And so this is from our textbook right here, but there's others. But you look at the first base, second base, third base, and then I'll tell you what the amino acid is in the protein. And so let's take a closer look at translation. That's occurring on the ribosome, and this is a kind of a cute cartoon of that. And it involves all 3 types of RNA. Transfer RNA is going to be bringing amino acids to the ribosome. Remember, the ribosome itself is made up of rRNA, and then messenger RNA is the genetic code itself. So the process of translation is where a series of codons in messenger RNA get translated into a polypeptide sequence. And so transfer RNA is crucial. Transfer RNA, sometimes it's called taxi RNA, which picks up a passenger, an amino acid that's in the cytoplasm. And you may know that either amino acids come from food, or sometimes the cell needs to make them if necessary. So ribosomes are going to add the amino acids together and make the growing polypeptide chain. So transfer RNA is a really neat molecule. We often think of RNA as single-stranded, but the truth is, since it's single-stranded, it can enfold in some interesting shapes, sort of like the letter L here. And in fact, let's see if I can draw on this, one side of the L is holding an amino acid that it picks up like a passenger. And then these 3 nitrogen bases on the bottom are going to temporarily bond with the codon of messenger RNA. Let's see if I can do this. So if the codon of messenger RNA is AUG, this is what would determine what amino acid it is. And then this would temporarily bond with it. This would, of course, be UAC. And then on messenger RNA, what we call is the codon. We call that 3 nitrogen base the codon. So we're going to call this the anticodon. The anticodon is on transfer RNA. Be more about that in a moment. And so up here again, transfer RNA, how big is it? It's not very big. It's about 80 nucleotides in length. Do you notice here it hydrogen bonds with itself, forming double-stranded areas. And so it was earlier described as a clover leaf, although our latest X-ray crystallography computer graphics show that it's sort of looking a little bit more like a letter L. And again, this is the part where it attaches to messenger RNA, the anticodon down here. And then this is the place where the amino acids are attached. Up here it sort of looks like a candle. So at the wick is where the amino acids attach. So the amino acid goes up here, the anticodon goes down here. And so each arriving transfer RNA at the ribosome is going to be carrying an amino acid, like arriving. And it's the anticodon that's going to allow it to temporarily attach to the messenger RNA. So this is complementary to the codon, the anticodon. And here is a sort of a cartoon of the amino acid. And again, transfer RNA, I can illustrate that. So up here is the amino acid, and then down here would be the anticodon that will attach to the messenger RNA. And so let me point out something. What's the point of this? The point of this is to make a chain of amino acids. And so you're not going to do it if you just have one amino acid. So you might imagine it's going to involve another transfer RNA is going to come in. It's going to be carrying an amino acid, and I'll just put a question mark. What amino acid is it? And so if you look down here and you say, well, I wonder what the codon is. Let's say the codon is CCC. So if you were looking through your textbook, if your textbook is open right now on your iPad, or if you're looking at a decoder, you could look at this and say, okay, CCC, the code for that is the amino acid, what? I happen to remember it. It's the amino acid proline. And so proline is going to attach. And so the anticodon that will allow transfer RNA to attach happens to be the opposite or complementary to CCC, which is GGG. Most important thing, I'm going to emphasize this right now, it's the codon that determines the amino acid. It's the codon that determines the amino acid. And again, what's the point? You want amino acids close enough together so that they can bond and form a peptide bond. That's what you're trying to do, is link amino acids together to form a polypeptide. So this is how the cell gets that done. And so what's interesting is tRNA it's made in the nucleus just like all the other types of RNA. But what it can do is leave the nucleus, go out into the cytoplasm, and it acts as a little taxi again. It picks up an amino acid, it delivers it to the ribosome, and then drops it off, and then goes out and picks up another amino acid, very much like a taxi cab. And so here it is again, transfer RNA, carrying an amino acid. Here's the anticodon, which is allowing it to bond to messenger RNA. And so what's fascinating is some of our most powerful microscopes can actually see ribosomes. These big black-looking raisin-like structures are ribosome. I don't know if you can see this. This little thread right here is the messenger RNA. The protein is so small you wouldn't be able to see it, but this is actually in the cytoplasm, messenger RNA being translated into protein. I find that that's pretty cool that we can take a picture of that. And so the ribosome, I emphasize the ribosome. The ribosome's a pretty big deal here because it's the location of where the protein is being made. And let's not forget that the ribosome itself, the reason it's called ribosome is that it's made up of ribonucleic acid and proteins. And so let's talk about that just for a second. Let's say that a ribosome, so one of these things is a ribosome. Each one of these is made up of many proteins, like this. These are proteins. Kind of weird that a ribosome makes protein and it's composed of protein. And then there's something called a small subunit. So this is the small one, and then a large one. So these proteins are produced, and you may recall this from a previous discussion. These proteins are produced on already existing ribosomes. They travel back into the nucleus, into a region called the nucleolus or nucleolus, and then they're wrapped with, yes, you guessed it, rRNA. So rRNA is RNA, single-stranded polynucleotide, but it doesn't carry the genetic code. It makes up the structure that is a ribosome. So this is one kind of RNA that makes up the ribosome. The other RNA that I was talking about just a moment ago is transfer RNA. Transfer RNA carries amino acids, and so that's its function. So you get ribosomal RNA, transfer RNA, and then everyone's favorite RNA is, of course, messenger RNA, and that's kind of linear in appearance, like this, with its nitrogen bases sticking out. But the truth is, all three of them are needed. Messenger RNA is needed because it carries the genetic code on how to make the protein. The transfer RNA is needed because it brings the amino acids to manufacture the protein. And of course, rRNA is crucial because it functions as the ribosome itself. So I hope you're appreciating that. It's kind of a cool point. So, I don't know, looking at this picture, I like it. Looking at this picture of a ribosome makes me think that this brown thing and this green thing and this pink thing are different, but in fact they're very similar. This is ribosomal RNA, this is messenger RNA, and this is transfer RNA. What's interesting is, do you see how sort of it looks three-dimensional here? Well, you have to picture this as being a three-dimensional structure, and this is like a little cave. The small subunit attaches to messenger RNA. Here's the codon. So if you have your decoder, if you look at AUG, which is often the very start of every protein, this is the amino acid met, and it comes in and it docks, parks right here. But it's kind of like these parking spots on the ribosome are analogous to like a white zone. If you're driving a car, you can only park there temporarily, you can't just hang out. So it hydrogen bonds, and then the next thing is another transfer RNA comes in and parks right here at the amino acid site. And then once there's two amino acids, yes, you guessed it, once the second one arrives, see there's a site called the P site where the protein will begin to grow, and then the A site is where the new transfer RNA comes in. So here comes the new transfer RNA, and when the new one comes in, the two amino acids, let me just sort of illustrate this. This is one amino acid, here's the R group, here's the other amino acid, and when you link two monomers together, you're going to create a dimer. So this is the peptide bond between the two amino acids, and then water is obviously produced every time a peptide bond forms because it's a dehydration synthesis. So the ribosome itself, and this is a crucial point right here, the ribosome itself is the enzyme, the catalyst for the formation of a peptide bond. I find that to be kind of cool, and it's because of its three-dimensional shape. Like for example, back here you could see the large subunit is what's really catalyzing the bond between the two amino acids. That's an important point. So I like this. It's a series of steps. So here's the first one. The second transfer RNA comes in, forms the peptide bond, and then of course the ribosome translocates and moves over, and then this tRNA goes out into the cytoplasm to get another amino acid. And then another one comes in, and another one comes in, and lo and behold, it's sort of like each time a transfer RNA comes in, it'll form a peptide bond until you create this polypeptide over here. And eventually, you might wonder, how does it stop? How do we know it stops? You may recall that once the protein is finished, there's 3 codons that are called stop codons. And these stop codons don't code for any amino acid at all, and that's how you know that the protein is finished. When the protein is finished, protein becomes detached, the ribosome breaks up into large and small units, and you're good to go for the next time around. So again, this is a real picture. These are real ribosomes making protein. The protein's too small to see, but here's a cartoon of it. So it shows it in steps. The large and small attached to the messenger RNA. Here's the protein forming like this, and then you reach a stop codon, and then boom, your protein is completed. And let me just sort of remind you of what that is. That could be anything. That could be, for example, an enzyme. And it's like the cell will only make that when it needs it. And again, so therefore, you're like, well, how do you control that? Well, then you just don't make the messenger RNA. And how do you not make the messenger RNA? You don't transcribe it. And so that's how cells regulate protein synthesis. And then this is a good illustration of how when the protein is being made, you finally reach on messenger RNA a stop codon, and there's 3 of them as I mentioned. That means the protein's finished, and then the ribosomes part their merry way. And so this is how the protein is finished. And again, this last statement right here is that the real functionality of a protein is not just its primary structure or its amino acid sequence, but rather how it folds and forms interesting shapes, which is the secondary and tertiary structures. And so let's go here, and I have a couple of animations that I want to show you. This is a pretty good one right here. Let me enlarge this and then get it to play if I can. And so here's the small subunit, and here is the large subunit, and it's attaching to messenger RNA. Here's the very first amino acid that is shown in red. This green guy is the transfer RNA, and just as you would predict, the codon over here is going to allow UUU is this orange amino acid, whatever it is, and then the peptide bond will form right there. Who's catalyzing it? The large subunit, and then that transfer RNA takes off, and then the next one comes in. And so, do you notice here on the P site is where the protein's growing? The A site is where the next amino acid is coming in. This is the A site. You read the codons 3 at a time, and see how the protein is elongating? So it continues and continues, and you make this big, big, big protein until eventually the protein is finished. And you will eventually reach a codon that is a stop codon. So the protein is cruising along like this, and then eventually a stop codon. This is something called a release factor. I'm not going to get into that kind of detail, but that's kind of cool. And then the protein is finished, and everybody parts their way. So that's nice. Okay, let's check out this. This is, I think, a really neat animation too. And so here is, as you can see, transfer RNA is being produced in the nucleus, or this could be messenger RNA. So this is the process of transcription occurring. So here's DNA, this big blue guy, little review. This big blue guy is RNA polymerase. And so this is how all RNAs get made. In the nucleus, there's new RNA nucleotides that are being transcribed. And so at this point, all these RNAs get into the RNA polymerase, and they're cruising along, cruising along, cruising along. And then you say to yourself, well, if this is in the nucleus, and I know proteins are being made in the cytoplasm, eventually that RNA needs to leave the nucleus. And so it reaches the termination sequence. You may recall that. And when it reaches the termination sequence, that means that the RNA is finished. So just want to point this out. All RNAs, ribosomal, messenger, and transfer, are made in the same way. They're just different genes in the nucleus. And so what this guy is talking about is it's sort of like transferring something like music from your hard drive onto a CD, or sort of copying it. That's what he's getting into. So here's the RNA, and predictably it's going to leave the nucleus, and how it leaves the nucleus is through the nuclear pores. And so now is where translation takes place. Translation is going to take place in the cytoplasm. And so there's the small ribosomal subunit, kind of cool, attaches, and then there's the large subunit. And so these little things sticking up right there are the nitrogen bases of messenger RNA. This is a really fast process. See these sort of like green ghosts that are coming in like this? These are the transfer RNAs, and the transfer RNAs carry these little red guys on the tips, and those little red guys are the amino acids. And as they come in and attach, you'll be able to see at the top of the ribosome, and this is showing that each transfer RNA is carrying a different amino acid, and remember there's 20 different ones. And so they're coming in, and it's really the messenger RNA codon, which is the code to make a protein. So if you can remove the large subunit and look inside, you can see the two of them attaching here. And so there's this red thing as the protein that's coming out. It's kind of neat. And so I really like that video. That's a good one. Let's take a look at this. This is good too. Let's take a look at this one first. This is showing the process of transcription that we just saw occurs in the nucleus, and then messenger RNA goes out in the cytoplasm, and that's where translation takes place. And so here's the genetic code on messenger RNA. So if you were to predict what's the first thing that's going to happen, well, you have a start codon, small subunit attaches, and then a transfer RNA is going to come in with the amino acid methionine. And so there's the anticodon that's going to hydrogen bond temporarily. So there's the first amino acid in our protein. What will be the second one? Well, if you look over here, it'll be GUA. And so then the large subunit attaches. Here's the A site where the next transfer RNA is going to come. So you look at your decoder right now and you think, GUA, what will it be? What will it be? You're like, I'm not sure. So you look at your decoder and you figure it out. And so the next one coming in is valine, and then it temporarily attaches. Now notice what's going to happen up here. The amino acids are now close enough together that the large subunit can form the bond, the peptide bond. Now that it's attached, this transfer RNA doesn't have it anymore, it takes off. Ribosome moves over, translocates, frees up an open spot, the next one comes in. What's coming in? CGG. So if you look CGG, it's arginine. Then here's the anticodon, which allows it to park, forms the peptide bond, and then here we go. We're cruising along like this. And so it's a series. And so an animation is really helpful. It's difficult to draw the steps, although it's useful to try to do it. So you cruise along, maybe the protein's thousands of peptides long, and eventually you'll reach a stop codon. And when the stop codon arrives at the site, there's this sort of release factor, but it doesn't, I'm not sure this is going to show that, but the protein is finished and then everybody parts its way. That's kind of a cool one too. And then finally there's this one. I find that this one is really, really good. I saved it for last. And so here's messenger RNA. It leaves the nucleus, and so now we're in the cytoplasm. These little green guys are the transfer RNA, the little purples are the amino acid. Here's the small ribosomal subunit. It attaches to messenger RNA. There's methionine attaching. There's the large subunit attaching. So this is the initiation. Now we're going to elongate and make the protein. There's the peptide bond that's going to attach. Notice there's two of them in the P site and the A site. Now it moved over. Now this is in the P site. So the A site's open. So the next transfer RNA comes in. How does it know to come in? Where's the genetic code? The genetic code is written in messenger RNA. So remember that. Messenger RNA is the genetic code. The green is just the anticodon, which will allow the correct transfer RNA to attach. And so once the tRNA is finished, it goes out into the cytoplasm and picks up more amino acids. Got to get those amino acids in the cell. Got to eat your protein. So when you eat your protein, all those amino acids get formed into your own protein. And so let me just sort of go to this picture right here. So this is what happens. It's quite amazing, you guys. It's the DNA makes the RNA is the intermediate, and then on ribosomes, the ribosomes are literally protein factories and they make polypeptides. So this is how cells make protein. I hope you enjoyed it. I hope you understood it. But more importantly, I hope you thought that the process was simply amazing. Thanks for watching.